This lecture is about writing a research protocol. This is lecture 7. What do we mean by the protocol? The protocol is a detailed plan of the proposed project. Why we need it? Because it provides written evidence of the need of the study and its feasibility. When we need it? Usually a research protocol should be written after proper and complete planning of the study. The benefits of the protocol. First, it's important to clarify the thoughts about all the aspects of the study. So it's important to collect all your ideas in this document. Second, it's necessary guide for working on the research. During your methodology, you need some guidance in order not to forget any detail. And finally, it's essential to get ethical approval and funding. When you need to get ethical approval, or if this study is funded by a body which is another organizer, you need to provide a document, and this document will be your protocol. Protocol formatting. The contents of the protocol should follow the following format. First, the title. Second, abstract. Introduction. Research question. Aim and objectives. Hypothesis. Method, which includes study design, setting, subjects and materials, sample size, procedure and intervention, timetable, source of funding, ethical approval, and then data analysis, further considerations, which may include pilot study, dissemination, and references, and finally appendices. These are the general formatting of the protocol. Some formatting may be included in each other. For example, the research question, aim and objectives, and hypothesis may be part of the introduction or can be separated like this. Starting with the title should be descriptive and concise. However, this may be the only part of the protocol that can be changed later. The name and affiliations of the investigators should be written below the title, like this. Abstract. The abstract should provide a quick summary of the project, should be concise, highlight the purpose of the study with a brief description of the methodology and data analysis. So the abstract just includes the objective of the study and the purpose with the methodology. There is no results, no conclusions. Usually it's between 250 to 350 words or no longer than 1 page. So you can see there is an objective, subjects and methods, but there is no yet conclusion and results. Introduction should explain the rationale of the study and refer briefly to the most relevant studies published on the subject. Research question should state the importance of the research question or research problem, or where is the gap in the knowledge, or why is the research is needed. Usually the research question can be written at the end of the introduction. As you can see here, you can just say that there is no study to date has investigated what you want to investigate. Aim and objectives. The aim and objectives should be clear and specific. The aim is the overall goal to be achieved, while the objectives are steps to achieve the aim. If you have more than one objective, you should state these objectives as primary and secondary objectives. But it's not favorable to put too many objectives that cannot be adequately achieved. This is an example of a study which aimed to compare the effectiveness of two types of orthodontic appliances. So this is the aim, which is the general idea or the goal of the study. What are the objectives? How can we achieve this aim? First of all, the primary objective is to measure the treatment duration. So this can be considered as a primary objective. Secondary objectives: first, quality of treatment using different indices to measure the outcomes, using incisor inclination and torque to compare between the two appliances, amount of anchorage loss between the two appliances, patient perception, and secondly, biological side effects of treatment by measuring root resorption. So you can see, the aim is the overall goal to be achieved, which is to compare which appliance is better, while objectives separated into primary objective, and the primary objective is the treatment duration. Secondary objectives can be divided into quality of treatment and biological side effects of treatment, and these should include how you will measure each one. So the objectives are the steps to achieve the aim. Hypothesis is usually stated as a null hypothesis, which means that there is no significant difference between the two types of treatment or interventions that you want to investigate. You can see here in this example that the null hypothesis stated that there is no statistically significant difference between the two appliances. This is the theory that we started with. Now, methodology. The methodology is the most important part of the protocol. It includes study design, which describes the study design that you plan to use when you're answering your research question. Setting means describe where the study will be conducted. Subjects and materials. Firstly, you should describe clearly the inclusion and exclusion criteria. How will the subjects be allocated to comparison groups? What are the materials or equipment to be used? Then sample size. This should explain and justify how the sample size will be calculated and what is the power of your sample size and your study. Procedure or intervention. This should include how the participants were informed and consented to participate if you have a patient participant. What are the methods of randomization and blinding if you apply this process or these methods? How and by whom the data will be collected? If you have intervention, this should include the proper name of the drugs and the commercial name, manufacturer, chemical composition, dose, frequency of administration. All these should be included if you have an intervention. If you have observation, for example, a questionnaire, this should be included and attached with the protocol. And if you have a specific laboratory procedure, this should be referenced adequately and include the reference Or if you have a modified procedure, this should be justifiable and included as well. Timetable should be included in your protocol, and this should schedule all the aspects of your research that should be produced. Stopping rules is important because it means what are the criteria of the study discontinuation. That means when you want to terminate your study if there is a specific side effects. So if one of your groups develop a side effect which cannot be tolerated by the patients, that means you should end your study even before it completed. Budget and funding. How much this study will cost and who will sponsor your study. This may be a self-funded study like most of your studies, or if it's funded by another person or another organization, this should be stated clearly. Ethical approval. This may include either the study will be submitted to ethics committee if it included human or animal participant, or if you have already an ethical approval, for example, for an ongoing studies, you should include this ethical approval and attach it with the protocol. Data management and analysis. It's important that prior to writing your protocol, to sit with a statistician and discuss the proposal of your project. What are the population and sampling, data collection, coding and analysis, and what is the computer program to be used for your analysis. Also, it's important to plan the data analysis from the beginning. That includes the descriptive and inferential statistics. All these should be clarified and outlined clearly. Analysis of assessing reliability should be included as well. Further considerations include pilot study. Sometimes you need to do a pilot study before your major study. So you can outline here if there is a pilot study will be conducted or will not be conducted. Then dissemination, which should outline the plan of your study dissemination. For example, if this project is an undergraduate project, or is it a postgraduate thesis, or is it a scientific journal article, or just for oral presentation. References. The protocol should be ended with a list of your cited references in the text. The reference style usually written according to the requirements of the institution or the publisher in case of an article. In case of your thesis, you should follow the requirements of the College of Dentistry. These are two examples of Harvard style and Vancouver style. For the Harvard style, the main difference between Harvard style and Vancouver style, you can see in Harvard style after the surname there is a comma, there is dot after each initial, and before the last name there is and. Usually the year written after the authors in case of Harvard style. Vancouver style usually used in journal articles. Sometimes you have at the end of the protocol appendices in which you should attach any additional materials, for example, patient information sheet and consent form if you have a patient participant, ethical approval if you have it already, questionnaires if you use a questionnaire, and etc. So these are the steps or the protocol formatting that you should follow while you are writing your protocol. It's important not to forget any of these during your writing. Now, what makes a good protocol? First, are the protocol format items that discussed earlier being addressed and matched with each other? So you shouldn't forget any of these items which discussed in the beginning of this lecture. Second, is it adequate to answer the research question and to achieve the study objective? For example, is your study design suitable to answer your research question? Third, is it feasible in the particular setup of the study? Is your study suitable with your time limit? Does it provide enough details that can allow others to do the study and compare the results? It's important that your methodology should be included in all the details in your protocol. Otherwise, others and you cannot follow your methodology properly. Pilot study. Sometimes you need to do pilot study. Why? First of all, it's always wise to pretest your protocol after developing it on a small sample to determine its feasibility and practicality and to identify any practical problems in implementation. That means, if there is any problem, it's important to identify these problems in the pilot study stage or in the protocol stage before you are starting with your proper study. If you find any weakness in the research process, this can be corrected in the protocol stage because this stage is flexible. And based on the outcome of the pilot study, the protocol can be modified before the study proper is implemented. Also, the pilot study serves as a training for the researcher and for standardization of the procedure of the research. All the measurements to be performed in the main study should be included in the pilot study in order to test these before you are starting. Also, using a pilot study may aid you to get estimates for the mean and standard deviation in order to calculate the sample size, especially if you don't have or it's difficult to find any other research to get these estimates from these. And inter and intra-examiner calibration can be used or done in the pilot study, so you can get your reliability tested in your pilot study before you are doing your main study. This is the end of lecture 7. Thank you.